Hello students, welcome once again to this channel. In this video we are going to see the part 2 of the lesson Sexual Reproduction in Flowering Plants, chapter 2 of CBSE class 12 biology. Come on, let us move on to the lesson. Gynosium. The gynosium represents the female reproductive part of a flower. If the gynosium consists of a single pistil, then it is called as monocarpellary. If the gynosium consists of more than one pistil, then it is called as multicarpellary. When there are more than one pistils, they may be fused together or they may be free. If it consists of a single pistil, then no problem. If it consists of more than one pistils, in some cases they are fused together, in some cases they remain free. Here we are going to see sincorpus and apocarpus. If the pistils are fused together, then it is called sincorpus. If the pistils are free, then this condition is called apocarpus. Here there is a diagram of pistil. In this there are three parts. Atop the pistil, stigma is there, then comes the style, then at the bottom of it we can see ovary, and then comes the thalamus. Now, parts of a pistil. The pistil has got three parts: the stigma, the style, and the ovary. The stigma serves as the landing platform for pollen grains. When pollen grains from the male reproductive part comes, they land on the stigma only. Then what about the style? The style is the elongated, slender part beneath the stigma. That's what we have seen in the diagram. The ovary. The basal bulged part of the pistil is the ovary. Unlike the style, the slender stalk, this ovary is somewhat bulged. The ovarian cavity, that is the locule, is present inside the ovary. The placenta is located inside the ovarian cavity. So, inside the ovarian cavity, we can see the placenta. The megasporangia arises from the placenta. So, where can we see the megasporangia? We can see megasporangia attached to the placenta. The megasporangia are commonly called as ovules. The other name of megasporangia is ovule. Now the ovary. The number of ovules in an ovary may be one, a single one. Example, wheat, paddy and mango. The number of ovules in an ovary may be many. Examples, papaya, watermelons and orchids. Now here comes a typical diagram of an anatrophous ovule. What is meant by an anatrophous ovule? Here the ovule remain upside down. Now let us see the parts of the ovule. Atop the diagram we can see the hilum, then funicle, then the micropyle, then comes the micropylar pole, and we can also see the integuments, the outer integuments and the inner integuments. Then comes the nucellus, then the embryo sac, and opposite to the micropylium we have got chalazial pole. Megasporangium, in other words ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. We have already learned that ovule is attached to the placenta. But how is it attached? By means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. So the place where the ovule is attached to the funicle is known as hilum. Thus, the hilum represents the junction between ovule and funicle. So, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. So, ovule is protected by means of a kind of envelopes called integuments. These integuments cover the nucellus except at the tip. So integuments cover the nucellus all around except at the tip. At the tip there is a small opening called micropyle present. So micropyle is nothing but a small opening in the ovule. Again, megasporangium, that is ovule. The chalaza is present opposite to the micropylar end. So opposite to the place where micropyle is present, at the other end we have chalaza. It represents the basal part of the ovule. So chalaza is the basal, the bottommost part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. So we have already come across the term called nucellus. Now we are going to learn what is meant by nucellus. Nucellus is nothing but a mass, a group of cells which is present inside the integuments. The embryo sac or female gametophyte is located in the nucellus. So the female gametophyte or the embryo sac is located inside this mass of cells called nucellus. An ovule generally has a single embryo sac formed from a megaspore. So generally an ovule has a single embryo sac. This embryo sac is formed from a megaspore. Megasporogenesis. Megasporogenesis. What is meant by megasporogenesis? The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. So, how are megaspores formed? They are formed from the megaspore mother cell. This process is termed as megasporogenesis. Megaspore mother cell is a large cell that contains dense cytoplasm and a prominent nucleus. The megaspore mother cell, which is abbreviated as MMC, undergoes meiotic division. So due to this meiotic division, meiosis, results in the production of 4 megaspores. So the megaspore mother cell undergoes meiotic division. As a result of it, 4 megaspores are formed. Female gametophyte. In a majority of flowering plants, one of the megaspores is functional and the other 3 degenerate. We have already learned that 4 megaspores are formed. Out of 4, only 1 megaspore is functional and the other 3 they get degenerated. Only the functional megaspore develops into the female gametophyte, that is embryo sac. So only the functional megaspore develops into a embryo sac, the female gametophyte. This method of embryo sac formation from a single megaspore is termed as monosporic development. An important term and also an important question based on this. What is meant by monosporic development? The method of embryo sac formation from a single megaspore is termed as monosporic development. The nucleus of the functional megaspore divides mitotically to form two nuclei. So, out of 4 megaspores, 3 get degenerated and only one is left, that is called functional megaspore. That functional megaspore divides mitotically to form two nuclei. The next topic, very important one, 8 nucleate embryo sac. What is meant by 8 nucleate embryo sac? It means an embryo sac which contains 8 nuclei. Now let us see how are these 8 nuclei are formed. Already we have learned that 2 nuclei are formed. These 2 nuclei move to the opposite poles and form the 2 nucleate embryo sac. So these 2 nuclei, they move to the opposite poles and form a 2 nucleate embryo sac. 2 more sequential mitotic nuclear divisions occur and form 4 nucleate and 8 nucleate stages of the embryo sac. So now 2 nucleate embryo sac is there. It undergoes a mitotic division and forms 4 nucleate stage. That means it contains 4 nuclei, and again it undergoes one more mitotic division and gets doubled and form 8 nucleate stage of the embryo sac. These mitotic divisions are free nuclear in nature. So all these mitotic divisions are free nuclear in nature. What is meant by free nuclear? The formation of nucleus without a cell wall, that is called free nucleus. It means nuclear divisions are not followed immediately by cell wall formation. It doesn't mean a cell wall will not at all get formed. It will get formed but not immediately. That condition is called free nucleus. After the 8 nucleate stage, cell walls are laid down, leading to the formation of the typical female gametophyte, that is embryo sac. So now, due to a series of mitotic divisions, now the embryo sac has come to a point or a stage which is known as 8 nucleate stage. It means the embryo sac has got 8 nuclei in it. After this formation, only later on cell walls are formed and it becomes a typical female gametophyte, that is embryo sac. Now, 7-celled embryo sac. What is meant by 7-celled embryo sac? How many nuclei are formed? We have learned that 8 nuclei were formed. By forming 8 nuclei, the embryo sac has become a 8 nucleate stage. Now, out of the 8 nuclei, 6 of the 8 nuclei are surrounded by cell walls and organized into cells. So, out of these 8 nuclei, 6 of them are surrounded by cell walls and they have become cells. The remaining 2 nuclei, called polar nuclei, are situated in the large central cell. They are situated below the egg apparatus in the central cell. Now, if you have a close look at this diagram, then you can understand very well. Now, this is called the 8 nucleate but 7-celled embryo sac. How come it is possible? Now have a look at this diagram. The pink color shaded ones are nuclei. How many nuclei are there? Atop the embryo sac there are 3 in the chelacal end, and again at the bottommost part there are 2 nuclei in the synergids and one more nucleus is visible in the egg cell. So altogether there are 6. Apart from these 6, there are 2 more nuclei which are located at the center part of the cell. So, as far as nuclei are concerned, there are 8 nuclei. So 8 nucleate stage, this one is. And when it comes to cell structure, at the chelazal end, there are 3 cells with 3 cell walls. And at the bottom of it, there are 2 cells inside the synergids and 1 more cell in the egg. So altogether, how many cells are there? 6. And there are 2 nuclei located at the central part of the huge big cell. So the whole diagram comprises 1 cell. So altogether 7 cells. So when it comes to nuclei, there are 8. But when it comes to cell, there are 7. That's why this stage is called as 8 nucleate 7-celled embryo sac. 8 nucleate 7-celled embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. As I had told earlier, how many cells are grouped together at the micropylar end? 3 cells are grouped together at the micropylar end and constitute the egg apparatus. At the bottom of the diagram, we have seen 2 cells are there in the synergids and 1 more there in the egg cell. So the egg apparatus consists of 2 synergids and 1 egg cell. So what is meant by egg apparatus? It consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus. What is meant by filiform apparatus? At the end of the synergid, we have got micropylar tip. At the micropylar tip, a special cellular thickenings are found. They are called filiform apparatus. Now, what is the function of this filiform apparatus? The filiform apparatus guides the pollen tubes into the synergid. When the pollen grain comes and lands on the stigma, it develops pollen tube, and the pollen tube grows gradually and reaches what? The egg apparatus. When it reaches the egg apparatus, the filiform apparatus is the part which guides the pollen tubes into the synergid. Now another 3 cells are located at the chelazal end. In the diagram we have seen it at the top of the diagram. They are called as antipodals. So antipodals consist of 3 more cells. Now altogether we have got 6 cells. The large central cell, the whole diagram itself is considered as a cell. That's what it is called as the large central cell. The large central cell has got 2 polar nuclei. On the whole, the embryo sac becomes an 8 nucleate but 7-celled one. Now the next topic is pollination. In flowering plants, both the gametes are non-motile. What is meant by motile? The capacity to move, mobility. So in flowering plants, both the gametes, both the male and female gametes are non-motile. They can't move on their own. So they have to be brought together for fertilization. So by some means, both the gametes should be brought together so that fertilization can take place. Transfer of pollen grain from the anther to the stigma of a pistil is termed as pollination. So pollination is nothing but the transfer of pollen grains from the male gametophyte called anther to the stigma of a pistil is termed as pollination. Now kinds of pollination. What are the 3 kinds of pollination? The three kinds of pollination are autogamy, Geitonogamy and Xenogamy. What is meant by autogamy? Pollination within the same flower. What does it mean? A flower consists of both male and female reproductive organs. In that case, pollination takes place within the flower. That is termed as autogamy. Then Geitonogamy, pollination within the same plant, not within the same flower. This takes place in the plant which consists of two sorts of flowers, two kinds of flower. One is staminate and another one is pistillate. What is meant by a staminate flower? It contains only stamens. What is meant by a pistillate flower? It contains only pistils. So in a single flower, we cannot have both reproductive organs. So in this case, pollination takes place from a staminate flower to a pistillate flower of the same plant, not same flower. This is termed as Geitonogamy. Then comes the Xenogamy, pollination in between different plants, not the same plant, different plants. In this case, the plants produce different kinds of flowers. In the case of papaya plant and all, we can see one plant produces only staminate flowers, not at all pistillate flowers. Then another plant produces only pistillate flowers. In such type of cases, pollination takes place from plant to plant, that is termed as xenogamy. Now let us learn in detail about autogamy. In this, the pollen grains from the anther are transferred to the stigma of the same flower. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity. So in order to autogamy take place, there must be a synchrony in between pollen release and stigma receptivity. That means the release of pollen and the receptivity of stigma should take place at the same time. Another thing is the anther and stigma should lie close to each other. In cleistogamous flowers, autogamy takes place. In what kind of flowers autogamy takes place? Cleistogamous flowers. Now let us learn what is meant by cleistogamous flowers. Chasmogamous and cleistogamous flowers. What is meant by chasmogamous and cleistogamous flowers? Chasmogamous flowers are open flowers. They are not closed. In these flowers, the anther and stigma are well exposed. They are exposed. They remain open. They are not at all closed. But cleistogamous flowers are closed flowers. They don't open at all. In these flowers, the anthers and stigma lie close to each other. As they remain closed all the time, in these type of flowers only autogamy take place. Some plants such as viola, that is common pansy, oxalis and common lina produce both the types of flowers, chasmogamous and cleistogamous. So it is a peculiar condition. In some species, as we have read it, viola, oxalis and camelina, they produce both kind of flowers. I mean the flowers which remain open and the flowers which remain closed. Now, cleistogamous flowers. Cleistogamous flowers are invariably autogamous, as there is no chance of cross-pollination. Why is it said so? We have already learned that cleistogamous flowers always remain closed. So there is no chance at all for cross-pollination. I mean pollen grains from other flowers to come and land on the stigma of this flower. It is not possible for pollen grains from other flowers to land on the stigma. So autogamy is assured. These flowers produce assured seed set even in the absence of pollinators. So in the case of these flowers, pollinators are not at all required. Even in the absence of pollinators, they produce a seed set. Then comes Geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant is called Geitonogamy. Although Geitonogamy is functionally a cross-pollinating process, it is genetically similar to autogamy. Hope you have got it. So why is it said so? Functionally it is a cross-pollinating process because the pollen grain comes from a different flower. So functionally it is functionally it is cross-pollinating only because the pollen grains, they don't come from a same flower. But genetically it is similar to autogamy. Why is it said so? Because though it comes from a different flower, the flower belongs to the same plant. So what about its genetic makeup? The genetic makeup remains the same. So even though the pollen grain comes from a different flower in Geitonogamy, genetically it is similar to autogamy because it's going to produce genetically similar offsprings. It is because the pollen grains come from the same plant. Then comes the xenogamy. Transfer of pollen grains from anther to stigma of a different plant. What about this? In this case only, the pollen grains come from a different plant, not from the different flower, entirely a different plant. In this type of pollination, only genetically different pollen grains are brought to the stigma. Yes, because as the pollen grains come from a different plant, so the pollen grains are genetically different from the stigma of a plant. So it produces genetic variations. So with this lesson, with this part of the video has come to an end. Let us meet in the next video. So don't forget to subscribe and share it to your friends, and also don't forget to press the bell icon so that you can get more and more notifications.
Like, share and comment and subscribe our channel and click the bell button for more videos.